Okay, so today I'm going to be talking about horizontal and vertical gene transfer. So with horizontal gene transfer, there are three different types of horizontal gene transfer. There is transformation, transduction, and conjugation. So with transformation occurs when a cell picks up DNA fragments from existing dead bacterial cells. So what I mean is when a bacterial cell dies, it'll leave behind those little fragments of DNA scattered throughout their environment. A new bacterial cell will come in, pick up those new DNA fragments. It'll go through homologous recombination. It'll go into the bacteria's chromosome, into a matching sequence or similar sequence. This new DNA could offer traits like antibiotic resistance, greater adhesion, ability to evade immune defense. There's a term researchers and scientists use when a bacterial cell has a higher affinity to pick up those DNA fragments, and that term is competence. So if a bacterial cell is more competent, it means that it has a greater ability, has a greater affinity in picking up those DNA fragments scattered throughout their environment. It's actually so crazy, in fact, that some cells will actually kill other bacterial cells just to pick up their DNA and incorporate and get new traits. There's a term for that, it's called fratricide, so it means killing one's brother. So think of bacteria killing another bacteria, they're killing one's brother. So they will actually kill bacterial cells and they will take their DNA and they will incorporate it because they'll get new traits from it. Transduction is a little bit different. Transduction occurs from a bacteriophage, so that means a virus that is targeting bacteria specifically. So the way transduction works is there's two kind of pathways or cycles. So in lysogeny, a bacteriophage will infect a bacterial cell with a viral genome. That genome will eventually work its way to the bacterial cell's chromosome. That viral genome within the bacterial cell's chromosome will then get, I'll draw it out for you, will then get replicated through many generations of binary fission. Essentially what happens is now you have all these bacteria cells with this viral genome, and then what happens is called induction. So induction will happen when a bacterial cell gets stressed or damaged, and essentially what it does is it kind of wakes up the viral genome, and the viral genome will end up cutting itself out of the bacteria cell's chromosome. Now we're in the lytic cycle. So the lytic cycle is when the viral genome kind of takes over the host cell's machinery and commands it to copy a bunch of this viral DNA and viral proteins, which leads to a bunch of bacteriophages being built. I'm sure you guys are familiar with phage capsids. So this is actually the outer protein that is also getting kind of manufactured within the host cell. The phage capsid is what's actually going to be surrounding the viral DNA and protecting it. And then in return, it's building all these bacteriophages that's eventually going to build up. It's going to cause the cell to kind of burst and scatter and explode with all these viral bacteriophages. So it's actually pretty crazy. It's like alien-like almost. You have all these little bacteriophages kind of bursting out of the cell. And that occurs in the lytic cycle. So transduction is when it's viral. It's when a bacteriophage infects a cell, that bacteria cell replicates through many generations of binary fission, and then eventually it will kind of disperse a bunch of more bacteriophages with that DNA. Conjugation is a little bit more simple. So conjugation requires the direct contact between two cells. So what ends up happening is a cell will make contact with another cell using their pili or pilus. The pilus will then retract, so it will kind of insert itself into another cell. It will then retract, pulling the cell even closer, and then a bridge is formed kind of through the pores or channels within the bacteria cell. This will allow the plasmid to then get copied and to then get transferred over via the bridge to the other bacterial cell. So conjugation is just the pili sticking into the cell getting closer, a bridge forming, the plasmid getting copied, still staying in the original cell, but getting copied and getting transferred out into a new bacterial cell. So now that new cell that didn't have a plasmid now has a plasmid. In fact, it also has the plasmid with a gene for a pilus or a pili. So in theory, if you have a couple cells with conjugation plasmids or pili or pilus or whatever, and it kind of spreads through a population, eventually that whole population will have plasmids that have genes that code for pili, and eventually they will all be passing genes and DNA through conjugation. Now onto vertical gene transfer. Vertical gene transfer is very simple. It's probably what everybody is most familiar with. Vertical gene transfer is just binary fission, mitosis, meiosis. It is just the passing of identical genes down from an older generation to a younger generation. So you have one cell, for example, if we're doing binary fission, we have one cell. That older generation will split into two cells. both with identical DNA and genetics. So prokaryotic cells do this through binary fission, and then eukaryotic cells do this through mitosis, meiosis. Yep, quick overview of horizontal and vertical gene transfer. So transformation is the uptake of DNA fragments scattered and left behind from dead bacteria cells. Transduction occurs when DNA is transferred via a bacteriophage. And conjugation occurs when plasmids or genes or DNA is transferred through a pilus through direct cell-to-cell contact.